as you can see from the title of this presentation, I'm in part reporting the first phase results of a research project. I actually have two audiences for this presentation. The live audience, if you can have live virtual audiences, is the DNA study group of the Castle Rock, Colorado Genealogical Society, and I'm involved in that study group. But also the second audience consists of the people, the researchers who are involved in the McCoy research project and other folks who may be following how we're doing. And so I had requested the ability to record the session anyway for those folks, and I'm glad we're going to be able to do it. I also have two purposes or two aims for the presentation. The first is to do that reporting of results. The second is to use that project as a vehicle for a pretty tutorial overview of Y-DNA analysis, what it is, how it works, some of the things you might hope to learn from it. And I'd like to make a request about questions. If you have questions during the presentation, and they're of the, wait a minute, I didn't understand that sort, I'd like to address those soon, but not if I was about to say that same thing. So give me a chance. If you see me leaving a subject and there's a problem with your understanding, feel free to ask a question. I think we'll ask that the questions generally be in the chat. And I have an ability to see it, but Chris is monitoring the chat as well. You can also put questions in the chat that are for the question and discussion section later. So, and we'll cover any that weren't covered on the way through. So with that said, our particular research project is trying to tie together several lines that are known or strongly believed, or were known or strongly believed at the beginning of the project, to be connected to one another. In some cases, we thought that because we had Y-DNA connections, in some cases because we had connections via traditional research. It started with my pairwise contact with two of the other people who were in the research group, and it gradually grew to include these 5 lines at present. I just put this slide up for completeness so that you could see, so that anybody who wants to see whether they might connect with the line or be interested in the line can see it. But this slide will be more informative for you in understanding what our situation was at the beginning of the study. We have 5 lines, and we know some or all of their, well, no, we don't know all in any case, but we know some of their descendants, and at least one descendant of each of these 5 lines is a participant in our study. They're all genealogists, active genealogists who are mostly working on their own lines, naturally enough, but who are interested in cooperating to try to find out how we're connected. And these lines are, these are the oldest or earliest well-documented ancestors in these 5 lines. 2 of them are known to have come from Scotland, this Donald McKay and Mad Donald McKay. These are all lines that are almost certainly descended from the McCoy clan in Sutherland in Scotland. So we want to find the ancestral connections. And basically that means we have a big blank up here. We know this much about us, about our lines. We want to know how those lines are all connected up here. They could all be 5 sons of one father. They could be pairwise, all different, going back in time. At the time we started this study, we had no idea. And under the circumstances, since we're talking about paternal lines, naturally Y-DNA was a reasonable place to start. And so that's what we did for our first phase of research, and that's what I'm going to talk about mostly. We also had one very specific hypothesis. This one arose out of a belief, my belief and Warren's belief. I'll tell you about the quotation marks in a minute. my belief and Warren's belief that our earliest documented ancestors, my McKinney and his Hugh, were probably brothers. They had similar migration paths, locations, family naming conventions, lots of reasons to think that they were closely related, and brothers seemed tenable. So we had the general goal and we had one specific hypothesis. So I'm going to now talk about Y-DNA, but I'm going to go kind of back to basics first. And I know you all know this, but just in case somebody actually doesn't, or in case anybody needs a refresher, all of us, if we are genetically, quote, normal, I don't mean as opposed to abnormal, I mean as opposed to, well, the next word I'm after is deviant. I'm getting myself in trouble here. But ordinarily, each of us has 22 pairs of autosomal chromosomes, or autosomes, that's just a short name for autosomal chromosomes. And those chromosomes behave in one way. And I know you guys have all been studying those. So you know that they segments of those chromosomes recombine, they swap around during meiosis, and various segments get sort of passed down a little bit at random to descendants. The sex chromosomes are different, both from the autosomal chromosomes and from each other. And we're going to be talking about the Y chromosome. Normal people, genetically normal people, have two X chromosomes if they're women, or one X and one Y if they're men. There are certainly other combinations possible. Biology is a messy business. And the Y chromosome is inherited strictly along paternal lines. So that's one important property that the Y chromosome has that no other DNA has. And it's a convenience when you're doing Y analysis, because you already know where up in your ancestral tree this came from. You're just trying to learn more about that path. The basic DNA building block for any of these kinds of DNA, not mitochondrial, but the autosomal X and Y, is the single nucleotide. And that's the first of a number of technical terms that I'm going to have to use, but you're going to hear me say over and over, don't let that term intimidate you. A single nucleotide is one place on the chromosome. That's all it means. However, if we called it one place, it would be very non-specific. You couldn't assume that something specific is meant by it. And a single nucleotide is a single location on the DNA strand, and it contains, as the molecule that's here, it's a little more complicated, but it's using a new phrase I just got tonight, that's more than you need to know. You can think of it as being one of four bases. Gosh, I'm losing the A. God, you know, normally I can do all of these. Adenine. Somebody help me here. I can't believe it.
Adenine, guanine, thymine, and cytosine?
Cytosine, I think. Okay. I blank on words. It doesn't mean I don't understand, although it could, but I blank on words all the time. Apologies for that, but we're all who we are. And so the presence of a particular one of those bases in a particular location can have very strong genetic significance if it occurs within a gene. And it can not mean a lot, but still be useful to us from the point of view of DNA analysis. And that's the case with most of the single nucleotides we'll encounter in Y-DNA analysis. I'll explain why. You're already aware, I think, that autosomal analysis consists mostly of identifying identical segments, hopefully long identical segments that you share from a DNA match cousin, and are able to verify via traditional genealogical techniques a connection back to a most recent common ancestor, and that can be very useful. But the way that it works is that you identify long sequences, hundreds, thousands, tens of thousands, I don't know how long they are, long sequences of adjacent single nucleotides that are identical. That's how autosomal DNA works. Why DNA is different? It's different part because we know our connections are along the paternal line, because we know that to a first approximation, Y chromosomes are inherited without change from father to son. They do change, or we wouldn't be able to use the technique, but they mostly don't. For that reason, for these reasons, the differences here that are meaningful to us are much smaller. They tend to occur in a single location along the Y chromosome, and the mutation rates here. There are two different mutation rates involved in Y DNA, but they both happen frequently enough to be interesting. Each of those two types of DNA, Y DNA analysis, I'll tell you what they are in a minute. Well, it's on the slide. Each of them tends to mutate at the rate of once every 2 or 3 generations. And that means that out of, for example, 23 million sites on the Y chromosome, one of them changed between me and my great-grandfather, or between me and my grandfather. But when you combine those two mutation patterns, it works out, the arithmetic works out such that there's a pretty good chance, I think more than 50%, that there is some difference in the Y chromosome between me and my grandfather, or certainly between me and my great-grandfather. So the mutations are meaningful in genealogical research time. We look at two different kinds of happenings on the Y chromosome when we do Y DNA analysis. One of them is short tandem repeats. Don't let that term intimidate you. It just means a little segment repeats over and over, one after another, like a tandem bicycle. And the other are single nucleotide polymorphisms. Well, that's just sites in which a single nucleotide can have more than one value in different people. They both mean very simple things, but because there's a need for technical terminology to mean specific things, the terms are often intimidating. I have in the past found the terms in Y DNA intimidating, so I speak from experience. Here's what short tandem repeats are. They're situations in which at one particular location along the Y chromosome, because we're talking about Ys, they also occur on autosomal chromosomes too. But in one particular location, there's a sequence such as guanine, adenine, thymine, adenine that repeat over and over again, one after another, some number of times. And during the process of inheritance, during the sexual reproduction process, the number of times that these repeat can change. Here's how I regard this is my non-technical, or I'm not a professional genealogist or geneticist, but my way of looking at this is that most of the material on our chromosomes is junk. It's not genes. It's not the stuff that makes us human, or determines what proteins will be produced, or exactly how we grow from an embryo, or those things. It's filler. And some of it's there because we've actually absorbed genetic material from bacteria that were with humans 2 million years ago. Some of them have real purpose, I think. And I think of this the same way I think of when our great-grandparents went to the post office and bought stamps. They got a sheet of stamps with no perforations, and they had to cut them apart with scissors. And that means space had to be left between the stamps so they could be cut apart without damaging the stamps, and without messing up the aesthetics of the situation. And I think that these repeats are a little bit like that. They're you can cut anywhere along this line and not cause trouble, but sometimes in the process, the number of repeats change. That's my cognitive model for this. If it helps you find, if you're a professional geneticist and you object vigorously, this is more than the audience needs to know. So that's what short tandem repeats are. They just little segments of single nucleotides that repeat some number of times. And so I may have 8 repeats and my grandfather might have had 9. Single nucleotide polymorphisms look like this. Here's the DNA double helix, and here are the bases, one on each side, that pair with each other and bind together the 2 strands of the double helix. And in a particular location somewhere along this very long double helix, there might be a different value, cytosine here and thymine, cytosine in this case, thymine here. And whenever there is a place, a nucleotide along the chromosome at which that can occur, with and the organism still be viable, when we learn about it, it becomes a single nucleotide polymorphism, a one place variable contents place. Didn't say it very well, but I think you see what I mean. So this is an SNP, single nucleotide polymorphism, because it has one value on this DNA strand and it has a different value, obviously for a different person on this one. Now I'm going to take you to FTDNA in a minute and show you how that shows up on the website if you're looking at what you see when you've done a Y-DNA test. But I had a hard time deciding in which order to do this, and I decided to show you the tests, even though you won't really understand yet if this is your first exposure, STR markers or SNPs in any more detail than what I've said. But FTDNA, family tree DNA, which is the primary vendor of genetic genealogical genetics, I guess, genetic genealogy, DNA tests that are useful in genealogy. Y, especially, I'm sorry, they're the primary one for Y, not for DNA in general. And they have offered in the past, these first 2 have been discontinued. They weren't very meaningful anyway, but they've offered very small tests when they first started that tested 12 sites at which these short tandem repeats could occur. And that was so meaningless that if I would get an email saying you have a match with 0 differences on 12 markers, I would throw the email away. There's no point in looking. That's not yet significant enough for me to even pay attention to. 25 was a little better. 37 starts to be interesting, mildly interesting. And they offered a 67. They now offer 37, unless it's changed recently, 37, 67, and 111. And those are all different sizes of analysis of the short tandem repeats. So this Y67 test, for example, tests 67 of those places on which one person might have 8 repeats and another person 9 and another person 10 or whatever. This is the current best test if you're doing only short tandem repeat testing. It tests 111 markers, and that's the kind of test that family tree DNA offers as a way of trying to get an idea of how closely you're related to other people who have done Y-DNA tests. But if you're going to get deeply into this, you really need to supplement that, not so much with 700 STR markers, but with a SNP, with an analysis that incorporates the SNPs. And you'll see why that is before we're done. So here is what you see if you have a Y-DNA sample at Family Tree DNA, you see when you go into your Y-DNA match page, first thing you see is that, please pay attention to this row right here. I think you can all see my cursor moving back and forth. So I could bring up this display for everybody who has 12 markers or more and with whom I'm connected, and that's 860, to whom I'm related closely. That's 869. Not very interesting though. I mean, some of those are way far removed on the ancestral tree from me. So here's for 25, 37, 27, and 10. Everything I'm going to be looking at, 111. Everything I'm going to be showing you is about 111, because our first step in doing our study was to bring one person from each of our 5 lines up to the best available test, which is not actually the Y111, it's the other one, the big Y700, which also includes SNP analysis. But here's what you can see here. I don't know if you noticed the names of our participants. Oh, I promised to tell you about the quotation marks. This, first of all, Rudy ought to be in quotation marks. I am the researcher who is involved in this study for our line. And I'm actually the organizer of this study. And whenever I show anything that's about autosomal DNA, it's my test that's being used, but I'm not in the paternal line of McCoy. So I have used a cousin's DNA sample. I don't have permission to show you his name. I just haven't reached in to ask him. I want to use my name anyway to keep things straight, because you're looking at all this information from, quote, my point of view, but it's really my cousin's sample. And Warren, I had in quotation marks because I haven't received permission to identify that person, who is a researcher in our study and whose Y-DNA sample we are using in the study. So for each person that I'm sufficiently closely related to, with however many markers I've chosen, I would get a different set of people over. If it was 67, it would include these, but it would include more and so on. But for all those with whom I'm related no farther removed than a genetic distance of 10. I'll show you what that means before we're done. I see a block of information about them. This is George. He's one of the researchers in our study, and he was on the far left of the graph I showed you before. And this says that he's done the 111 big Y1, big Y700, which includes not just 111 markers, but 700 or so. And we have a genetic distance of 1. That's very close. And it's, although we don't know who our common ancestor is yet, it's a little surprising with a genetic distance that close, but we don't. And on a set of 700 of them, 607 of them, of which yielded valid tests for both testers, where we have only 2 differences. Talked about haplogroups later. His earliest known ancestor in his line is Donald McKay, born 1800 in Scotland. So that's the information you can see from this particular page. And here's Ian Stewart McKay. His is another of the lines. He's deceased, but 2 of the genealogists of his family are in our research project. And here's Warren. He's again, he's on the 3rd line in that very first graph I showed you with Hugh McCoy, 1792 North Carolina, and so on. So this is kind of information that you see. And if I click on George's name here, these of course are PowerPoint slides. I can't click on it, but I can push the space bar and you'll see what I would see if I did click on it, which is his profile page. It doesn't have a lot of information. It does, of course, have an email address for him. And that's true for everybody who has a Y-DNA sample at FTDNA. They don't all work. There's somebody I'd love to reach who used his Comcast email address. And by now he's probably moved away and isn't using Comcast anymore. And in any case, the email address doesn't work. So can't count on that. But There's an email address and his earliest known paternal and maternal ancestors, and both Y and mtDNA haplogroups. And again, we're not going to talk about, we'll talk about Y haplogroups, not the rest, and surnames. That's kind of the collection of information that is easy to see directly within the web pages that are produced by FTDNA, the site. There are naturally enough ways to go and look at your own sample in more detail and do some other interesting things. But the real meat of analysis in a study of the sort that I'm reporting here, it is in the surname projects. First time I saw this spreadsheet, which stretches off 100 more columns to the right and many hundreds of columns down, and it looks very complicated, I was really intimidated by it. I'm sure you're not, but I was, and I'm going to decode it for you. So I know they like to think you're intimidated by it, but you won't be by the time we get done, hopefully. But I like to show you a little bit about it before I simplify it. I'm going to, in a minute, start throwing away parts of it we don't care about. But I know you can't read it very well, or I suppose you can't, but each line here, each of the white or gray lines that are running across here represents a single Y-DNA sample, or putting it differently, a single person who was the test taker, the tester for that sample. And there's information from here over to here, I can't point at it exactly, about that person or DNA sample. And then there's information from here across another 100 columns about STRs, short tandem repeat values. And going down, there are also colored headers here, and each of those represents a group of relatively closely related testers. This grouping is not automatic. It's done by the moderators is the wrong word, but anyway, the folks who manage the particular projects. This is the McKay, I guess you say, surname project. It's on FTDNA, but it's a separate project, and there are hundreds of these. Some surname, some location, some special interest. And if you have a Y-DNA sample at FTDNA, you would be well advised to join some and perhaps all of the projects that are available to you that appear to be relevant to your particular DNA sample. Certainly, if you're lucky enough to have a surname project, especially one with this many participants, you may get a lot of information out of it. So I'm going to simplify this one step. I'm going to, by pure chance, the very second group here and the third group here are our groups. We're right at the top. Ignore this first group of 2 people, but the next 2 groups are ours. So I'm going to simplify it by throwing away everybody but those 2 groups, and even a couple of those. I have reasons more than you need to know. I'm going to eliminate all but 6 samples from the first group and 3 from the second. And I decided to include this. I'm showing you now all the way across to the right-hand edge of the table. It won't fit on here without my breaking it in 2. But I want you to get a visual feel for what 111 markers is before I throw away a bunch of those. So here we are with 9 Y-DNA samples, all done at the highest available accuracy, content, which is the big Y700 test. And 5 of them are our 5 lines. That's where we stand now. That's partly, as I say, because we did targeted Y-DNA testing to get us to that point, so we could do what I'm about to show you. I'm now going to simplify this by throwing away, it should be clear to you now, I think that wherever there are changes in the numbers of repeats of these short tandem repeats, they represent a mutation, using the word mutation a little loosely. They're representing mutation that occurred between a father and a son. And those happen fairly infrequently. I already mentioned that. Well, every 2 or 3 generations on one of the 23 million, no, sorry, one of the, 700 sites of this sort along the chromosome that they're studying now. So most of these markers are going to have the very same value all the way down the table, certainly all the way down in our 2 groups. And those are not the ones we care about. We care about how many we differ on and what the differences are. So for the next slide, I've eliminated all of those STR markers on which we are all the folks in these in these 2 groups from these 9 samples have the same values. I've only retained those in which we're different. So now we can look at the situation in a little more detail. To make it easier for me, but I think for you as well, I'm doing the whole presentation from the point of view of my line. So I've taken my line, which is here, so that's me, Rudy, and it runs across here and picks up again where I put my name again. I've taken it as the reference point, the reference sample for this purposes of this discussion. So when I color green, anything that's different, it means anything that's different from my line. And my line, therefore, has no green colors in it. Let's look at George's line. He was the one I was closest to when I showed you the Y-DNA matches display from a family tree. And he has only one difference. So let's see what that means. You can see here that on an STR marker called DYS 712, my line has 21 repeats, 21 times that some short sequence of bases repeats all together in succession, tandem like the bicycle. And George's line, or George's sample, I should say, to be accurate, has 22. That's a difference of 1. You might remember that I pointed at where it said genetic distance 1. This is what it means. Let's look at the line of Ian Stewart McKay here. Let me go up here. Let me run across the top here. I should have done that before. So here's Ian's line. This is his sample number and FTDNA. Here's the surname that it represents. I've added his name to show you what in this table means what. His earliest well-documented ancestor is Donald McKay, also called Mad Donald McKay, and an interesting guy about which books have been written, who was born in 1753 in Scotland. He died in Canada in 1833. And this is his Y haplogroup. Again, you got to wait. We'll get there. So here's George's line, similar information, and mine, and Warren's, and Bruce's. Now, Ian has one difference from my line. Has 3. Here's 1, 36 to 37. Here are 2. Actually, he has 4 differences. Well, they count this apparently as a genetic difference of, oh, I'm sorry. My line is 16. It is 1. So he's off by 1 here, 36 to 37. He's off by 1 here, 17 to, here's my line, 16. And he's off by 1 here, 22 versus my 1. That's a genetic distance of 3. And Warren has a different distance of 6. Here's 1, 2, 3, 4, 5, 6. So, and Bruce is farther away. I forget. I think he's at a genetic distance of 10. So you might suppose, just on the basis of this much information, that of these 5 folks, George is my closest relative. Ian is next, then Warren, then Bruce. That turns out not to be exactly right. You have to regard the the genetic distance from STR analyses as an approximation. But there's a way to get much closer, and that's through SNPs and haplogroups. I'm hoping that's what comes up next. Oh, I just put this slide in to remind you. I just showed you George, distance of 1, Ian, distance of 3, Warren, distance of 6. Hopefully you now have a visual point of reference so you understand what those things mean. And those numbers have nothing to do with SNPs or haplogroups. They have to do with a genetic distance based on STR markers. So now we're going to proceed with SNPs, single nucleotide polymorphisms, and you've seen this graph before. But now I'm going to assign it a particular meaning because I'm about to build a tree for you that's based on a particular understanding of a transition. Excuse me. So let's assume that this is the DNA strand for somebody we'll call father. This is his son. They differ across their entire Y chromosome, only in position 4. I didn't count very well. This one here is hiding, but I'm going to call this 1, 2, 3, 4 for convenience. And in position 4, dad has a C cytosine, and in position 4, the son has a T thymine. There's also a color coding, although there's not a formal standard for how the colors apply, that the colors, when they're used this way, tend to have the same meaning by everybody, I think, but that doesn't matter. What matters is this diagram shows a transition in one single nucleotide polymorphism, that is to say at one such location on the Y chromosome, from cytosine to thymine, from father to son. So now I'm going to build a little tree for you. Initially, it's going to look a little funny because you can't see what much of it is, but I'm going to call this block for the moment block 1. That's H1 here. And I'm going to greatly simplify the single nucleotide polymorphisms. I'm going to claim that this block represents a C. Well, for the moment, it represents C, cytosine in position 4, and T, thymine in position 6. And for the moment, we're going to ignore all the other SNP locations. In a minute, we're going to say, well, it really means that they're all the same, except for these one different. But for right now, this is father. He has a C in position 4 and a T in position 6. Now, he has a son who has a mutation in location 4. It's just that same transition I showed you a minute ago. So unlike dad, in position 4, he has a thymine base, and dad had a cytosine in position 4, but they're identical on 6 and really on everything else. So that defines a new group, because this is a tree of groups, not of people. I know you're used to seeing ancestral trees where this descendancy relationship represents people, but I can break you of that habit here, or that understanding, by saying, let's suppose that father here has another son who inherits an exact copy, that's actually the most likely thing, of dad's Y chromosome. He doesn't appear as another block down here. He's in this group because this block represents the group of everybody who has this exact complement all across the Y chromosome, well, the SNP sites on the Y chromosome values. It's a group. It's actually called a haplogroup. Another technical term. Don't let it bother you. Haplo means simple. Haplogroup means simple group. It doesn't have to mean very much. It's been adopted as the technical term in genetics for this kind of a group. That's what's important. So it's a grouping of everybody that's alike on this particular metric, the contents of the various single nucleotide polymorphism sites. let's suppose that, and by the way, haplogroup 1 here has everybody in it who is a son of a son of a son of a son, as long as they kept inheriting dad's Y chromosome. So this can extend down any number of generations. This could be what's called Y Adam, the root of the Y haplotree. This is called a haplotree because it's a tree of haplogroups. And there is an Adam, although it can be a moving target. You'll see how we might draw inferences about distant ancestors based on current Y DNA testers in a minute, but we can gradually derive an ever more precise understanding of the whole tree, all the way back to some earliest person we know about right now from our available information, and that's called Y Adam, and that's the reference against which all the other haplogroups are judged, and they form a tree. So let's assume that the son, that's dad, remember, that's son, he has a son. He may have a whole lot of sons, and they have his very same Y chromosome, in which case they're all part of haplogroup 2. But this son has a different value in location 6. He's gone from thymine to adenine, and the location and SNP site 6 of the Y chromosome. So that forms a tree. Now, so far, you've been looking at that tree from the top down. This is how it formed. This represents the history, the ancestry, old to new. But there's another way to look at it. We can start with current Y DNA testers. We know their values for all these SNPs. And so we can draw inferences about it. If A and B are both in this haplogroup, it means that they match on all but these 2 sites. Site 4, sorry, they match on all. It means that they match on all the sites, including these 2 sites. If C has one difference from this haplogroup 3 and is otherwise the same, and A and B have that, don't have that difference, you can infer that A and B have a common ancestor, and that ancestor and C have a common ancestor. You can draw inferences about backward from the current testers about the shape of this tree. And that's why SNPs and haplogroups are powerful, because they can actually tell us the pattern. Certainly, well, we'll cover the uncertainty in a minute, but they can tell us the pattern of our Y ancestry more exactly than the STR markers. I told you that according to the STR markers, my closest relative within the project is George, and next was Ian. That's not the case, as you'll see in a minute. So I'm going to show you how FTDNA presents this information for us. But in order to make it easier for you to see, I first want to show you a different way of displaying a tree. Here's the sort of standard tree diagram that we're used to. Here's a diagram which FTDNA calls a block tree. Mathematicians call it an icicle diagram. They have other names too, I'm sure, but I think that's the common name. These are the same. Watch and I'll show you. G is above F and D. G is above F, oops, sorry. And G is above F and D. F is above E and D. F is above E and D. E is above A and B. E is above A and B. So just different ways of showing the same structure. But you might pick one over the other depending on what it is you're trying to emphasize. If you want to emphasize the structure, this is better. If you want to put a lot of information in the blocks, these little tiny blocks don't work very well. So FTDNA has adopted this, and they have actually done some other fancy things and built a display which I consider brilliant for showing this stuff to us. It's a little busy. I'm not going to show you everything it can do for you, but let's look at what it tells us that's about the lines in our study that helps us understand these lines. First of all, it says your branch. So my branch, my cousin's branch really, but I called it Rudy, is right here. And it has an exact match. That is to say, from the point of view of SNPs, that's George, who's also in our study. And we together, we two, and nobody else yet, form a haplogroup called, instead of 1, 2, and 3 like I was using a minute ago, it's called RFT200202. This is a haplogroup with only 2 of us in it at present. That's because, of course, only 2 of the Y-DNA samples that would have detected it have yet been analyzed and have given permission to be included publicly in the McKay research, McKay surname project, or maybe other places where we might find this information. One of them says their ancestor was in Scotland. I should change this to show the US. It's going to turn out that when we find the next guy in the line that it's from Scotland, but I haven't proven that yet. Be back with you in a second. So here is Warren. Warren is in the next haplogroup up. You remember just a minute ago, this is George, this is me, this is Warren. He's in the next haplogroup up. Now back to the block diagram. This is the haplogroup number. He also doesn't know his earliest ancestor and mine were both born in 2 years apart in North Carolina. So he probably ought to have a US flag here too, but neither of us ever thought of it. And here is Ian, who had a genetic distance of 6. We're 1, this is 3. He was 6 on the STR markers, but really this is where he fits. This is where his haplogroup fits. And here's Bruce, the last of our 5 lines currently in the study. So what can we conclude that's helpful? This kind of analysis is by no means magical. It doesn't, there's one sense in which it doesn't tell us a lot. It certainly isn't going to point directly to our remote ancestors or any of that, but I find the things that we can learn from it very useful. So here's the first thing. You've already seen this, but now putting it on the diagram that shows our 5 lines, what we've demonstrated is that George and I, or his Donald and my McKinney, have an ancestor. I'll call him ancestor A, have a most recent common ancestor, if you like, and who is common to us. And we both and Hugh have a common ancestor. I'll call him ancestor B here, but we now know that he is at least 1 generation farther back, and that ancestor A is one of his descendants. And Ian, or Mad Donald McKay, and all of the 3 over here, and these 2 ancestors all have another common ancestor, 1 or more generations farther back. And we know that Bruce's line has another ancestor guaranteed to be also an ancestor of all of us. We know that much. Now, that's useful all by itself. But also, we had a specific hypothesis for the study that McKinney and Hugh were brothers. You can see that they obviously are not. That changed my priorities because I was going to look for them as brothers first. And now that's not the first thing I'm going to do. First thing, I'm really going to prioritize finding ancestor A because that's the most practical next step, I think. But some other interesting things can happen. We have started actually phase 2 of the study. And I'll show you some things we're doing there. First of all, we're doing detailed descendancy analysis of these earliest known ancestors in our 5 lines. The reason for that, and there are multiple reasons, but the main reason is if we can bring one of these lines down far enough, we may establish autosomal connections if we're lucky. How far down do we need to bring the descendancy? In my opinion, we need to bring those trees down to everybody who was born about 1960 or before. That's a lot farther down than you normally think. But the reason is a lot of, by no means all, but a lot of, well, most of the people who are doing DNA samples on these genetic genealogy sites are not genealogists. They probably don't know their family tree back 12 generations. And a lot of them, maybe still most, are doing it to get the ethnicity information. And they may not really know much about their family tree, but they probably do know the names of their parents and grandparents. And if we're lucky and they put that much information in a family tree that they tie their autosomal sample to, and we build our descendancy tree down far enough to connect, they will become a verifiable DNA matches and known cousins. And that's very powerful. Once we have a sufficient pool of autosomal connections, well, you know, you have lots of DNA matches on Ancestry. Once we have enough known DNA matches via this method, we may find in more than one of them who have put or can be persuaded to put their autosomal samples on a place like MyHeritage. And once you do that, you acquire the ability to do segment matching. And to give you an idea, in one of my lines, I found 2 people I could verify via Ancestry. I do most of my work first on Ancestry. I found 2 DNA cousins that I could verifiably regard as DNA cousins descended from a known common ancestor and verified by traditional genealogical means. And I found, well, I have way more than 2 of what I've said so far, but I found 2 of them on MyHeritage. And that gave me segment information, which in turn gave me 70 more DNA matches that are probably descended from that same ancestor. Now, it all has to be demonstrated. You have to watch out for matches on the other chromosome, maternal versus paternal of mine. But I've already, in this case, done that because I had matches on these segments from both sides, and I got enough information to be able to tell the difference directly from just looking at the segment data. So that's powerful, because once you have a set of 70 or however many the number happens to be of people for which you have segment matches, you're rolling. So that's one reason that we're doing these descendancy analyses. Now it happens that we've started doing these descendancy analyses, but in one line, this line, Mad Donald McKay, there are 2 genealogists in that family who have done a lot of descendancy work already. And they're nowhere near down to 1960, but I used their trees to build a private searchable tree of my own on Ancestry. I put myself in that tree and each person in my line back to my earliest known ancestor in the line, and artificially, it's not real, artificially tied him, McKoy, to Mad Donald McKay. And I picked up 3 autosomal matches. That is incredibly encouraging for this reason. It means that all of these lines are connected within genealogical time. That's really lucky. Hang on. Here's why that's lucky. When you find haplogroup patterns like this, very often there are 2 or 3, could be 12 generations in this gap. And another 3 to 5 in this gap, and so on. The fact that we've established autosomal connections from here to here to here, I'll do it again in case you looked away. The fact that we've established autosomal connections that run from here to here to here means there aren't 12 generations hiding in here. There can't be very many hiding in these 3 gaps taken together for us to have autosomal connections. Now, they're weak connections. I think the biggest is 22 centimorgans and one is 11, but they're through multiple sons of ancestor C, multiple children, not sons, of ancestor C. And we already know via Y-DNA that this pattern of relatedness exists, and I'm pretty confident that those are real autosomal connections. So that's a result from the very beginning of work in what I call phase 2. Other things that we'll be doing include really working the timelines for these earliest ancestors. I doubt any of us have done the most serious kind of timeline for that ancestor. We need to really work those timelines and look for the commonalities. By the way, another comment back here. Another reason I think that this is not sorry, where is it? Well, another reason that I think this connection goes all the way from here up to there and back down is there are no common places here. These guys are all in different places, and all of that, except for McKinney, except for these 2 lines, they're in all different places: Scotland, Canada, Kentucky. Our lines are in North Carolina and Georgia and Oklahoma and Texas and stuff. So because they're all in different places, I don't think these autosomal matches are from lines other than the McCay lines, but could be wrong. So we'll do timelines. In the process of doing these detailed descendancy analyses, we may find individuals alive today who could therefore do DNA samples or have done them, whose DNA information would in some way or another be useful in helping us ferret out the situation we have with these 5 lines. There's an interesting case study of a family named Parker in the book Advanced Genealogical Techniques. I don't remember the next thing, something and case studies. It's a case study of the Parker family that used a lot more testers, more lines, a lot of targeted Y-DNA and targeted autosomal DNA to connect to discover their ancestral trees with a lot of accuracy, and being able to point to Larry Parker here, who obviously is the one who had the mutation in this SNP in many parts of the tree. So it can be done. I don't think we're either planning to spend the 30 years or I don't think we're that ambitious, but targeted testing may still be useful to us. We'll see. And additional lines may be helpful. I already know about 2. There's a line of in FTDNA in the McCoy surname project that's closer to me than these 2 lines. I think it's next. It fits right in here in terms of STR marker genetic distance. But I can't get in touch with this guy. He's the one with the Comcast email address. I think I may have figured out who he is, and I'm going to keep trying. He didn't return my first attempt to contact, so maybe it isn't him. But if we could figure out who this is and he wants to cooperate, we might gain more information. And there's a line of McCoy's, Henry Thomas McCoy, about whom information is available in various sites. And I have an autosomal connection, but no idea how it connects. So if I can find some descendant in his line who's tested or willing to do the serious testing, and in general, I think project members are willing to pay for people's testing if that's a hurdle. Then we might have another line from which we can gain information, and others will come along. I'm satisfied. So in conclusion, Y-DNA analysis was first of all, the obvious first step for us, because we're a paternal line study. But it was a good way for us to get ourselves organized and oriented with respect to our various lines. It helped us reject a hypothesis we will eventually work, but might have spent time on. And it is helping us see how to prioritize our future work in the study. And that's it.